to see if it's on site or online before returning back to them. So Dr. Ansia will talk about the genome assembly of the indigenous African plant species, Helichrysium. Dr. Ansia, are you ready to speak? All right, okay, here you go. I am gonna just spotlight you. I hope I can do this. Um, so I think I can spotlight you. All right, I can't spotlight you unless you start your video. So you wanna start? You wanna put it on so people that way I can spotlight you? Is that okay, Ansia? You're on mute, Ansia, if you're talking.
Sorry, I'm
Ansia.
just getting my stuff ready.
All right.
I wasn't ready to present right now. Let me just switch my lights on. Is it all working now?
Yes, we can see. Let me see if I found you again in the. So, okay, I need to spotlight you. All right. Okay, yeah. I think you're now part of the spotlight. Yeah, perfect. So share your screen.
Sorry, my stuff is just jumping around. Okay. So, hi everyone. I'm Antia van Koller. I am a senior scientist and bioinformatician from the South African Medical Research Council's genomics platform. And today I'll tell you a bit about the genome assembly that I've been working on for a complex African plant known as Helichrysium odoratissimum. So the name of this species is based from the Greek word helichrysum, and this is because of the golden yellow flowers that is characteristic of this genus. And the word odoratissimum refers to strong odor. That's because this is quite a fragrant plant, and that's why it's also known commonly as African incense. And in our native languages, it also goes by koichut or impepo. So this plant grows quite abundantly throughout Southern Africa, and it is one of the most popular traditional remedies. It grows abundantly everywhere, in the highlands, in the little slopes next to the road, not just in South Africa, but also in Mozambique, Zimbabwe, Malawi, and a little bit more north as well. It is of great cultural significance in South Africa. So parts of the plant is routinely burnt to rebuke evil spirits, or it can be inhaled for good luck or good fortune. It also has lots of medicinal uses. It's been used for the treatment of heart disease, chest pain. It has calming effects. It's also been successfully used to treat coughs, fevers, urinary tract infections, which suggests that this plant might have some antimicrobial properties as well. And again, the calming effects of the plant has also been used to treat things like psychosis, and this could be due to the presence of phytocannabinoids, which has been identified in other Helichrysum species. It's also used commonly as an insect repellent or perfume because of the strong odor, and it is one of the most frequently harvested and traded plants in South Africa, and it has a great potential for commercialization. So unfortunately, no fully assembled genome currently exists for any of the Helichrysum species, but there have been some studies on the karyotype and the genome sizes. And these studies show a lot of variation in genome size, chromosome number, and ploidy level. Multiple different levels of ploidy have also been reported within a single species, suggesting that this is an important evolutionary mechanism for this genus to drive diversification and ecological adaptability. So because there's no nuclear genome assembly available for any of these Helichrysium species, we then sought to sequence and assemble the genome of Helichrysium odoratissimum. And today I'll be presenting those results to you. So just a little background, because I'm sure not all of you are working with genome assemblies every day. So obviously a reference genome is very helpful for enhancing genomic research. But if there is no reference available, we need to construct it in a de novo manner. So after sequencing, this process relies on alignment tools that can align all the reads to each other in order to identify overlaps to make contiguous sequences referred to as contigs. And the overall goal is just to create one long contiguous sequence that encompasses the entire genome, but that's not usually how it goes. So you're more likely to end up with little fragmented bits. And you can do the process of scaffolding, which allows you to pull individual contigs together. But yeah, overall, this can be a quite computationally intensive process. And things like long read is really helpful for long read sequencing is very helpful for de novo assembly because it's a bit easier to resolve repetitive regions and inverted regions, which might struggle if you only have short read data. So we got a fresh plant and we were able to extract high quality DNA and RNA. And then we performed ONT sequencing on our Prometheon P2 solo sequencer, and we did the Hi-C and RNA sequencing on the MGI DNBC G400. These are just my colleagues that were working on this. So this is the overall plan for the assembly. We used the ONT data and I did the super accurate base calling to ensure that we got the best quality of data out. And this was followed by QC using nanoplot, trimming of adapters and filtering based on read quality. And we also used ONT data to try estimate the ploidy and the genome size of this individual. For the Hi-C data, we also did QC and of course the basic trimming and quality filter to make sure that we're only working with the best data. And I then used Helichrysium to do a de novo genome assembly, which integrates both ONT and the Hi-C reads. And the Helichrysium, the newest version, works really well with ONT data. I also took the error-corrected ONT reads to redo the ploidy and genome size estimates, just because it would be a bit more accurate with the error-corrected reads. After I had the draft genome, I integrated the Hi-C data again by doing alignments and lots of pre-processing to find the Hi-C contacts that would be informative of the 3D structure of the genome. And I used that to do the scaffolding using YARS. This was followed by lots of assembly QC and manual curation of the genome to ensure that everything is in the correct order. And then afterwards, we will do annotation. So some studies have been done on this exact species, Helichrysium odoratissimum, and it showed that there were both diploid and hexaploid individuals within the same species, and this was dependent on the location where the plants were. So smudge plot analysis showed that this individual is hexaploid, and the genome scope analysis agreed with this, and you can see the 6 peaks with the KMA coverage. This also revealed to us that this genome is highly heterozygous, so have about 5% heterozygosity, which is quite high. I think anything above 1% would be considered slightly more complex than normal. And so, yeah, and with quite a large genome size with up to 750 megabase per, megabase per haploid size. So this is the the Hiphyism command that I used, and of course referred to the ONT flag to indicate that this was ONT data and integrated the Hi-C data. I specified that there are 6 haplotypes, and that's why it produced assemblies for the 6 different haplotypes. Obviously, the higher the ploidy level, the less accurately Hiphyism can split the haplotypes, but it does improve the quality of the primary assembly. So for my further downstream analysis, I looked mainly at the primary assembly that was generated, which is essentially a collapsed version of all the haplotypes. And then I also looked at haplotype 1. So you can see there's a lot of contigs in this, so it is still quite a fragmented assembly. And there's some difference in the total size between the primary assembly and the hap 1. But overall, not a bad assembly if we look at the N50 and all the other metrics. So due to the very large number of small contigs that we found in these assemblies that we couldn't integrate elsewhere following scaffolding, so I did a lot of trial and error, we decided to just extract all the contigs that were smaller than 50 kb, and then we compared these sequences to known mitochondrial and fluoropath databases. And then if more than 50% of a contig matched to these database, we would remove that contig prior to scaffolding. So yeah, I just show for the primary assembly and the filtered assembly, I removed, you can see quite a lot of smaller scaffolds were discarded, and it didn't affect the total length much, or the completeness of the genome. So in the bottom here, I also have the BASCO score. So these are pretty complete assemblies, although there is quite a bit of duplication, but this could just be a normal observation for this species. So we integrated the Hi-C data. Now I think tomorrow there will be someone presenting on the details of Hi-C library prep and how this works, but just for a for a quick summary for those of you maybe not familiar. So Hi-C is a high throughput technique that is used to capture 3D chromatin conformation. So we know inside the cells, the DNA is folded in on itself, and through a process of cross-linking, we can ligate ends of the DNAs together that are in close proximity. So you can end up with DNA fragments that are not necessarily linear as they should be, but rather contain information from different pieces of DNA that are in close proximity to each other. And then once you have these fragments, you can prep and sequence the library as you would any other DNA library. And you can then generate a contact map like this, where you can see which regions of the genome interacts with the other regions. So our Hi-C data was really good quality, and from the pre-analysis that we did, or the pre-processing, I could see a really good capturing of chromosomal interactions with really good resolutions, with detecting differences at various interactions, various different distances. So this is um, the assembly metrics before and after scaffolding for both the primary and the haplotype 1. And you can see that the contigs did reduce in both cases, and you can see that the N50 significantly increased. My favorite part of these tables to always look at would be the L90, which in essence is the number of contigs that make up 90% of the genome. And you can see that has been reduced to 24 in the primary and to 34 in the HAP1. So a majority of the genome is made up by a small number of contigs, which is a good sign. So the next step is then the genome curation process, just to make sure everything is in the right order, and then we can assign the chromosomes. So the process of doing this, you would take the scaffolded assembly and generate a TPF file, which dictates the order of the assembly. And then you would also realign the high C reads to the assembly in order to generate that contact map, which you would then manually rearrange and assign chromosomes. And then you'd combine all that information. In the end, you would end up with your curated assembly. So how the manual curation process works. So sorry, this is not a great picture, but I think it's very useful. So when you generate the high-C contact map, you can see how different regions of the genome interact with each other. And you can see, for instance, in this example, if there's a little spot here, it indicates that that region of the genome likely belongs over here. So the manual curation process is basically just manual splitting of scaffolds and rearranging it until it's as meant to be. And here is an actual image of my high-C contact map for haplotype 1. And you can see that there are some interactions here off to the side, which we can then manually incorporate these scaffolds into these bigger scaffolds. And even if I zoom in on one of these scaffolds, you can see even within a scaffold, there is some rearrangement that needs to happen. But of course, this is a very complicated plant. It's hexaploid and it has a lot of repetitive regions, so it does make this process a bit more difficult, but we are gaining some momentum there. So yeah, our next step is just to finish off the manual curation of the genome by rearranging the high-C contact map on a pretext map. And then we can define the chromosomes and we can then export the data to generate the final assembly, which we'll upload to NCBI. And then we will do some genome annotation using the final assembly and the RNA-seq data that we have, so we can finally see, what is it about this plant that makes it so interesting. So yeah, just like to acknowledge everyone at the SAMRC and of course everyone at AfricaBP that have been working with us on this. And yeah, thank you for the opportunity to present here, and thank you all for your attention. Does anyone have any questions?
Okay, thank you, Ansia, for your presentation. Do we have any more any question on the chat box or on the Zoom call before we move to the on-site participants? So someone is saying, Mary Emaragi. Mary, do you want to unmute? So Mary is asking, I'm just going to read out her chat here. Let me see. So she's asking why chromatin rearrangements. Not quite sure the context of. Mary, do you want to provide more context to your question? So I'm just gonna. I think you should have. You should be able to unmute now and ask your question.
Thank you. Can you hear me?
Yes, go ahead. We can hear you.
Yeah. I know that I have not much knowledge about genome assemblies, but chromatin, like it looks to me like, are you trying to associate parts of the genome to see which ones are related? And what was that? What did that do to assembling genome? What are you doing? What's the strategy here? Thank you.
So if you're only using DNA sequencing, I think nowadays the assembly algorithms are very good where you can get a telomere to telomere assembly using just, for instance, very long ONT or PacBio reads. But for some of these species, it's a bit more difficult. Um, when you just use the whole genome sequencing, you end up with very fragmented genome assembly. So because the assembler is trying to, build the puzzle to put the assembly in order, but it doesn't necessarily know which order it is in. So if you combine the whole genome sequencing data with the Hi-C data, which captures the chromatin conformation, you can associate the different contigs with each other based on their spatial proximity. So because the Hi-C process ligates pieces of DNA together that are, we assume, within the same chromosome. And the closer they are to each other in the chromosome, the more intense the interactions will be. So based on the chromatin conformation, we can generate those contact maps to see exactly where the genome is, like the exact order of the chromosomes, or at least to see which chromosomes go together, and then we can define them based on their lengths. I hope that answers your question.
Thank you, Ansia. So I hope that answers Mary's question. Hewan, Dego, please, you can unmute.
Thank you very much, Doc, giving me the chance. It is a wonderful presentation. I learned a lot because it's new for me, the plant. I think it is African indigenous plant. Like one of my questions is like why it is really selected. Is there some kind of different variation or medical aspects? If you just briefly explain for me that. And then when I just wanted to know, because there is a number of ploidy in African plants, so have you ever seen some kind of repeated elements in the genome? And are there any ploidy events observed in this genome? And that is some of my question, but now I will read your papers if it is published, and I will look for more exposure in the future. Thank you.
Thank you. So this species was selected because it is of great cultural significance in South Africa. It is one of the most commonly used plants for traditional medicine as well, and it is expected that it likely has some compounds that are antimicrobial and have calming effects, maybe like the phytocannabinoids, which can have good effects with like inflammation, etc. So yeah, we are hoping to, annotate the genome to see if we can find the molecules responsible for this medicinal effects that the plant has. And in terms of ploidy, yes, so it is known even within the species that there are different ploidy levels based on the area in which the plant grows. And we do suspect that this could be due to just like a whole genome duplication that has occurred. So in that case, you would see a lot of duplicated regions of the genome, and this is something that does commonly occur in these plants with their large genomes. Um, it's not this is not published yet, but we are hoping to publish this soon.
So thank you, Ansia. We have another question from. Looks like you have a lot of questions, Ansia. We have one question from Laide, Laide Rasaki. Laide, do you want to ask your question orally, or shall I read it out over here? So he's asking, I am curious to know how you name the chromosomes if there is no reference genome already. So I think this is, yeah, there's a context already. Answer, you can answer this one. Right?
So we would name the chromosome based on the high C contact map. We would be able to see which regions interact most with each other, and so based on that, you can define the chromosome. But there's also some previous studies where they do like flow cytometry and stuff, where they can estimate the number of chromosomes that are present. So we'll then see if what we can identify based on the high C contact map and how that corresponds to the information that is known about like the number of chromosomes, for instance.
Okay, thank you, Ansia. So I think we're trying to go back to the on-site participants. There are some questions there. So they lost connection, so I am trying to get them back up. All right, Julian, I am going to make you a co-host. So Professor Julian, do you want to start your video? And then are there any questions on site, please?
Yes.
So do we have any questions?
Thank you very much for this nice and insightful presentation. You made mention along your presentation that this plan. Yes, we have a question here.
Please go ahead on site. I'm just seeing the control video now.
Presentation that this plant has an insect repellent cell insect. Am I clear?
Yeah, go ahead. Please carry on.
On mosquito. So I'm thinking of a way that this gene is responsible for producing strong odor is amplified. Meanwhile, the enzymes strong odor are amplified such that we can make it
So I think we could barely hear them. Ansia, are you hearing anything?
In much quantity, so that it can be used for commercial activities and to reduce.
So we could barely hear the control. So could you please type your
question? I said the.
Yeah, could you please type your question? Write down your question and send it to the IT or the control, and then they'll paste it in the chat box so we can access it.
Make the question clear again.
Okay, all right. Go ahead. Okay, go ahead. We can hear you now. We can see you rather. So we're trying to see if we can fetch the question from the on-site participants. Control, do we have the question now? Do you want to paste it in the chat box so we can read it out to Ansia?
An insect ability. Were you able to identify that gene responsible that? Can you share with us interest and your contribution? That is, I want to have an input, your input personally, on how we can amplify that gene so that it will be of a commercial interest, or it can be used to produce. It can be used to to produce something that will repel insects, that mosquitoes particularly, because that is where I have an interest.
Okay,
I don't know
answer.
if you
Did you get his
my
question? He's talking about the
Is it
translation.
clear?
Yeah, I think we got your question, though it wasn't so clear, but it's fine. So he's talking about how do you get the gene and then transform it, and then translate it to an actual thing that you can use. So if you can just, within the context of what you heard.
Yeah, sorry, I didn't hear the full question, but I think it was relating to how would you identify which genes may be contributing to specific effects and how that could potentially be commercialized. So this would be hopefully identified during the annotation process, because we also have RNA sequencing data. So we can use the genome and the RNA data to try and identify all of the genes that are being expressed, and then we can compare that based on homology to other plants to see if we can identify things that have been expressed in other plants that have had, medicinal effects, etc., and see how ours maybe differs from that. But yeah, it's most likely going to lead to additional studies to see if those genes that we'd identified are the ones, contributing to the effect that we're trying to look at. And then if it is established that there is a link between specific genes and a specific molecule that is produced for whatever effect, whether it's, insect repellent or something else medicinal, then that could potentially go into the commercial space where they can try to make that into a different project. But that's potentially not something that I myself will be doing, because that's not my field of expertise. So I'll leave that to the biotech experts.
Yeah, thank you, Dr. Ansia Vancola. Do we have any more question on site, please? Question on site? Any more question? Ansia, it looks like you've got lots of comments. Question. Sorry, do you have any more question on site, please?
No question.
All right. All right, thank you so much. We have another question here. It could get lots of feedback for you, Ansia. Looks like, yeah. So Matthew Moses says, please, my question is more like a recommendation. I'm a graduate student of medicine and surgery. I'm keen. Okay, I think kindly highlight what I need to have or know to succeed in this field. So I don't know if that relates to you, Ansia, or not, but you can just, this looks like more like career kind of stuff. So over to you.
I mean, I don't know if I have all the right answers for that one, but yeah, just see if you can get into a program where you can do research. I think you're in a good space at the moment. You can see who's working on what, and if there's anything that you find interesting, just reach out to those people, and yeah, just put your passion out there and make it happen. Good luck. And yeah, thank God for the opportunity to present here today.
Yeah, thank you, Ansia, and thank you for coming much earlier to present 1 hour earlier so that we can be able to align with other people who are not here. Hopefully they come in much later. And yeah, and hopefully we'll see you again in during the East and Central Africa workshop to talk about the genome. Thank you so much. All right, perfect. We're going to go to the next presenter. Again, you're supposed to speak in the next session, but you have to bring him forward. That would be Michael Landy, Kofi Landy. Michael, if I'm just gonna make you the co-host and please unmute yourself. Michael is at the International Institute of Tropical Agriculture in Kenya, and is also a PhD student at SLU, Sweden. He'll be talking about the genome assembly of the white star apple as part of the African BioGenome Project. So, Michael, do you want to unmute yourself, please?
Yes. Can you hear me okay?
Yes, please, we can hear you.
Perfect. Hi everyone, my name is Michael. Just allow me to put off the video just in case I experience any internet connectivity. So we'll start by sharing my screen. Okay. Mm. Can you see my screen now?
Yes, please, we can see your screen.
The whole, okay, perfect. So let me
Yes,
just
carry
get
on.
the...
Yeah, carry on.
All right, so as thank God mentioned, I'm based in Nairobi, Kenya, and today I'll talk about the African star apple genome. But for this talk, I will not talk about the introduction of this crop. I will dive into the genomics. We will have, I think, tomorrow 2 talks on library preparation and also propagation of this plant, this fruit. But as you can see, it looks a really delicious plant, and I'm really looking forward to visit Nigeria to test this fruit, the star apple fruit. When we think about plant genomes, we think about stories that we either talk to our children, and when you read a story from a book, that will be probably the genome, and the pages would be sort of the chromosomes, and the pages that you read will have the instructions that probably have you think in the genome world will be the genes that sort of give us the instruction, the functionalities of our plant, and why we care about plant genomes is these are just examples of the applications of plant genomes. And just to mention a few, if probably you want to think about crop improvement and breeding, sort of looking at wild type plants and maybe transforming them to domesticated plants. Obviously, plant genomes come in handy if you're thinking about resource conservation and ecological resources. I think this is probably the main theme of this workshop. So genomes come really in handy. If you're thinking about plant evolution and classification, genomes also are really, really important. And just to mention about epigenetics, thinking about beyond genetics, then you have to also understand the genome to explain any traits that are influenced by epigenetics, or rather the environment. So in the world of genome assembly, I think probably Tasian already mentioned, but think of it as solving a puzzle where you have little pieces that you try to join together to have a bigger picture, and that is the genome. And just to give an overview of genome assembly is these small pieces in a jig puzzle would be your short or long reads. So small pieces of DNA. I'll explain how they're produced in the coming slides. And as you're solving this puzzle, you tend to join these pieces, and these reads, you join them to form contigs, sort of bigger, smaller chunks of these small pieces. And these contigs you can also assemble them to scaffolds, and later on you have a full representation, for example, a chromosome, and within a genome, say, for example, with the star apple genome, you'd have all the chromosome, and then you can define actually the functionality of a plant from a plant genome. But when we always do this, the process of genome assembly, there are always some problems that we encounter while assembling these plant genomes. On this plot here, what you can see is, if you imagine this a plant is a diploid inbreed species, where you expect two copies of the genomes coming from the two parents are homozygous, then it's always easier to represent this genome as single haplotype reference assembly. And by haplotype, I mean one copy of a diploid. So a diploid will have two copies. So one copy will be the haplotype 1 and haplotype 2. And for similar copies, sort of homozygous genomes, you only need to have a single haplotype reference. And this is pretty straightforward. But when we have cases where we have outbred assembly, then it becomes a bit tricky. And you have, so here in gold and blue, these are contigs, or sort of a chromosome coming from one parent, so the maternal and the paternal. And always when you're assembling these pieces, you sort of don't know where the pieces are coming, either from the father or the mother. And you can always represent this either as a collapse representation, and how we call this we call it as haplotype mosaic reference, where you represent this as combined if you cannot really differentiate which contig is coming from which parent. But in the recent times, we do have some well-refined sequencing platform that I'll mention in the coming slides, where you can really define where contigs or scaffolds come from. And then you get a haplotype first reference assembly, where you know that, okay, this haplotype 1 is coming from this parent, and this other one is coming from the other parent. And this is specific for diploid, an example of diploid genomes. And so, as I mentioned, the long reads sequences, and here I specifically mentioned the hi-fi reads that are making us plant genome assembly to be really easy. And the fact that, and I will not get to the details of how this process goes on. I think we'd have a presentation on library preparation and get to understand it more. But just to point out here, the hi-fi reads have very high accuracy, and accuracy of more than 99% accuracy. And with this accuracy, you have the advantage of accuracy and also having long reads. And if you have these two combination, then it's really easy now to assemble a genome and to have really high quality genome assembly. So just to jump in on the workflow of the genome assembly of this species star apple, and just to mention here in green, these are the steps that I will share. So once we have this hi-fi reads, we do the quality control, because this organism has not yet been sequenced and assembled. It is always nice to estimate the size of the genome and also know the ploidy level, how many copies of this genome in these plants. And I will share some of the assembly statistics after doing assembly using this tool called hi-fi SM, that I will share the different steps. And then check for duplications and remove if there's any duplication rates within the genome scaffolding. And in the end, in red here, these are steps that we are ongoing. And I need to mention that this work is ongoing. So some of the results are preliminary results that I will share. And so after scaffolding, normally what you want to do is you want to do manual curation. You want to gap fill and remove any contaminations that are present in the genome. Evaluate the assembly, and then obviously at the end you want to look at the gene repeat and the repeat annotations within the genome so that you can explain some of the functionalities of this plan. And so the data that we use in this case is the PacBio Hi-Fi reads that I mentioned are really good quality. And we sort of use additional information that is the OmniC. These are the reads or data that we use to do the scaffolding part of the analysis. As I mentioned, the quality is really good. And here I'm showing, so the first plot is Hi-Fi reads, and in the bottom is the OmniC reads. And here is just the length of distribution of your library. And what is interesting to note, we always have reads that are way too short and reads that are way too high. And the first thing what we need to do is to filter out those extreme lengths. And what we did is we filtered the Hi-Fi reads based on the length of 10 kilobase pairs and to 30 kilobase pairs. And for the OmniC reads, because these are short reads, and so we only filtered any read that is 50 base pairs and above, and that's what we kept. And here I don't show, but we filtered out some bases upstream, so 5' upstream, around 15 base pairs. And for the OmniC also, we trimmed 14 base pairs upstream that were a lot of biases. And what I mean by biases is we couldn't, from the quality control, you can always see unproportionality of the bases within your library. And so we ended up with a total number of reads for the Hi-Fi reads, 3 million, and the length, as I mentioned, 10 to 30 KB. And then this was the also total length of the reads for the OmniC that we take to the downstream analysis. Now, for the first step is to, once we have our data, our clean data, what we want to try to answer is what then is the estimated genome size and what is the ploidy prediction. So what you see here is a plot from a tool called GenomeScope. And here, what you're seeing, these are KMA plotted. So this approach will try to estimate the size, and here it tells you the estimated size of this genome in one copy is around 822 megabase pairs. And you could already see that is a very highly homozygous genome of 99.7% and very low heterozygous level of 0.3%. And already here you see like the first peak will be your heterozygous KMAs that are pretty low from the percentage. And the second peak are homozygous KMAs that shows this genome is highly homozygous. And we used other tools called SmartPlot to just predict the ploidy prediction. And as you can see, so we use different types of KMA sizes to do the prediction. And as you can see towards the end here, AB, it estimates 82% that this genome is a diploid genome. And if we also do another trial with 31 KMA, then we get 78%, which sort of these two approaches tend to confirm that this is a diploid species, and the estimated haploid size is 822 megabase pairs, and it's really a highly homozygous genome. So after this prediction, we went ahead and do the genome assembly, and here I'm just showing the method Hi-Fi SM. We used additional two other methods to assemble these reads, and I will share the statistics, but just to focus on Hi-Fi SM because this was the main output that we take for the assembly. And what happens here is you have your input hi-fi reads. And as I mentioned, you'd have sets of reads coming from here in orange is from one haplotype, and then here in blue is from the other haplotype. And so this region here is an example of an heterozygous variant. And in the green, these are homozygous reads, so reads that are present in both haplotypes. And so what the tool does is first it will check for any sequence error and correct this, and will identify sites that are actually the heterozygous alleles. As you can see here, there's AAA, and from the other haplotype is CCC, which is the different allele. And so after it corrects, it sort of starts to build a graph of this genome, and these bubbles here are where we define reads coming from different haplotypes. And so once you get to these graphs, you can either go two way. Either you get the primary assembly construction where you only have the primary with one of the variant and have sort of some alternative assembly, or if you have other complementary data, and these are actually reads coming from both parents that help you to separate these two faces, which we didn't have for our case. And so we ended up representing this genome as a primary representation. And also the fact that it's highly homozygous, then getting the primary assembly of the genome was an ideal approach. And so after doing the assembly, we now did some statistics, and what you can see here is a screenshot of the statistics of the genome. So the first row is outputs, the primary assembly of HiFi-SM, and HiCanu, which is another tool that we use to assemble this genome, and file is another tool that we use. But just to draw attention on three key characteristics. So first is the number of contigs that the assembly generates. So you can already see around 7,111 contigs are only for the hi-fi reads. So what you'd expect is you want a lesser number of contigs. So the HiCanu and Fly tend to have a lot more contigs of the assembly. And in terms of the total length, well, this was really surprising because we already saw the estimated genome was around 820 megabase pairs. But here we are seeing a huge number of total length. So this is 2.4 gigabytes. Even more for HiCanu and Fly. Fly got 15 gigabytes, and this was 2.8 gigabytes, which is really, really huge. And also another factor to check is the N50 value, which was really good for HiFi-SM, 1.3 megabase pair. So this tells you the contiguity of the assembly. And for the HiCanu and Fly had very low, and this sort of already showed that these two assemblies here were highly fragmented. And that's why we move forward with HiFi-SM assembly. So then with this primary assembly, we went ahead and checked for the completeness of the genome. And tools like Busco will tell you how complete this genome is. And you can see here it's 98% complete, but then the duplication rate is really high, 87.6%. So this was really puzzling for this genome. So what we do next is we look for regions that are highly duplicated. And what we use is we use a tool called PatchTubes that has two main steps, where the first part, what it does is it takes your long reads and then maps it to the reference that did the primary assembly. And what you get is sort of a coverage output. And then the next thing, what it does, it splits the assembly into chunks of say 10,000 base pairs, for example. So split assembly by a certain number of length, and then it sort of does a self-alignment, right? So using the self-alignment and the coverage results, it checks for haplotypes, or rather contigs that are identical, and filters this as exact matches. And so it removes this duplicates this way, and you'll end up having a paged genome, and some of the contigs that were actually removed. And what is interesting with this tool is you can redo these 3 steps until you get a decent haplotype set, haplotype set that is either less duplicated, and it's also close to your estimated value. So we did this step, and then we moved forward and now get the statistics of the draft assembly. And here is the same that I mentioned before. So we have the number of contigs. So the first column is the primary assembly, and then the first round of paging, so the page up, and then the second round of paging. And you can already see in the total length from 2.4 gigabytes, then we get to a close estimate of the genome, which is around 821. And in terms of the N50 value, the last column, as you can see, is 3.1 million, which is really improved contiguity as compared to what we had in the draft genome. If you look at the duplication level, so for the second round of paging, we have a 6.6% duplication rate, which is actually more expected if you compare with even other genomes that have already been assembled. So this is really good. So we move the next step where we do the scaffolding using now the OmniC reads. And for the statistics again for the star apple scaffolds, we now have 596 number of scaffolds. The length is 821 megabase pairs, which is really what is close to the expected size. The N50 is 57 million, which is highly contiguous, and this was good. And already with the L90 value as 13, this sort of gives you an idea of the number of chromosomes that you'd expect for this species. And normally it's always +1 or -2, +1 or -1. And just here to show is a contact map showing the scaffolding of this genome. And the next step, what we do is then to do the manual curation of this genome. So once we've done the scaffold, so we need to manually curate this genome. And then because we have the scaffold, so we expect to have gaps that we need to fill and check for contamination within the genome. And we will submit this genome to NCBI, and later on do the genome gene annotation, and just to have an idea of the repeat content of this genome. And a point that I didn't mention is having the write-up and submitting it to be accessible to everybody. And just to conclude, so we expect to achieve a very high quality of star apple genome, and this is the best, the fact that we already have high quality reads both on the OmniC and the HiFi reads, and this would actually be relevant in the genome-assisted cultivar breeding to provide a better understanding of important metabolic pathway. And we will actually see some of the benefits of this crop in the coming days from the other presentations. And we would also use this data to do some capacity development where we will do some training workshop on genome assembly and annotation to build regional capacity and promote the intercontinental transfer of knowledge. And now I'd like to acknowledge everybody who's involved in this project, the Inkaba, who did the sequencing, and we will hear about them in the coming session. African BioGenome Project, led by Thank God, for actually organizing the whole workshop and also managing the whole analysis. IITA, where I'm based with my supervisor Andreas, and the team from ANCAB and IITA who have really assisted in making this a success. So thank you very much, and I'm happy to take any question.
Well, thank you, Michael. This is this is very detailed and good to see the progress. So we're just gonna, if you could please stop sharing your screen, Michael, so we can have
Yes.
the two screens together. We're gonna take questions. So do we know if people on site, do we still have people on site? Control? Is Control still on site? Are they still online, or just trying to see? Oh, it looks like we lost the on-site people, the connection. So we're going to take questions from those on Zoom, the participants on Zoom rather. If you do have any question, please use the icon to raise your hand or put that in the chat box, please. So we have some questions here. So Hawan, do you want to ask your question orally to Michael, please? Hawan, let me try and find you. Yeah, and when you can unmute now and speak, please.
Okay, thank you very much. I was watching and listening and learning a lot, specifically this star genome plant is really has a reference genome. It's because this is my childhood fruit, which I only know eastern part of Ethiopia. And it is really great resilience, and the wood is really great. But I didn't find that no research at all. And recently we are collecting this germplasm to disseminate to the northern part of Ethiopia, because as you can see, in the northern part there is a lot of degradation, and the forest is shrinking. But this is a fruit and by itself, and the wood is really great. And I'm so much happy to find out, lastly, the Africans are really sequencing this genome. So my question is, when you are doing the reference genome, as you know, the distribution of this plant is in the equator, I think around that, and a little bit to the north and a little bit to the southern part of Ethiopia. No, to the southern part of Africa. So when you are doing the reference genome, which accession or a variety have you used? And is it like, a kind of, can we trace the plant passport data? That will be really interesting. Then it will help us to proceed with our research in this plant too. Thank you very much.
Okay, that's really nice to know that the fruit is, you grew up taking the fruit. That's really nice. So onto your question, I'm actually really looking forward to actually test the fruit itself. Yeah. So to your question, so I think the accession was from Nigeria, but I think we will get more of the presentation on propagation of this plant tomorrow. Yeah. So maybe then we can have I think the accurate accession of this plant. I don't know if thank God you know, because I have no idea.
Yeah. So Hewan, this is from the IITA collection, the accession, and the presentation on the wet lab and the sample collection and processing part of this work will be presented tomorrow by the IITA scientists and by the genome facility where it was done. So you will get more background information on that. But definitely, it's the collection at the IITA. It's been there for a while, and then yeah.
Thank you.
Okay. Yeah, it's
Yeah.
good to see that you like the fruit as well, right? I'm sure many people on the call. So for many people who perhaps cannot map the common name to the English name, also this is agwalumo or udara, for instance. For many people who know the local names of the species, so it's hawked everywhere on the streets in West Africa. And as you just rightly also pointed out, Hawaii is also found in Ethiopia, which is good. Okay. Thank you so much, Michael. We have an additional question. Professor Anemugai has a question, and you want to go ahead, please?
Yeah, I think for me, I just want to appreciate the two scientists because it's showcasing the fact that we can do this work on the continent. My question to both, very briefly, you've been able to present very high quality data. It'll be interesting to hear about the start and whether it was as easy as you have projected it to be. What are the challenges, for instance? Just off the cuff, what are some of the challenges you faced? Was it the sampling? Was it the isolation of the nucleic acid, or what specific challenge did you have? Thank you.
Maybe we can also spotlight Ansia here. So it looks like this is a question for both of them. So Ansia, if you want to put on your video or something, so I can spotlight you if your video is off. So I can do it and then you can go take it back as well. So you want to put on your video, Ansia? So that I can spotlight you and yeah, okay, found you. All right. So this is more of a question. There's an important question, Professor Mugai, on what it is. So I'm sure you both heard about it. So you want to just talk about it, and I'm sure that would also help inspire others. Because one of the things AfricaBP aims to do is to sequence the genome locally. No matter the challenges, no matter anything, it has to be local. And of course, many people already have seen that. So let us know in the next 5-10 minutes. I want us to be very detailed, explanatory, and yeah, just say over to you.
Ansia, do you want to start or I can?
Sure, okay.
Yeah.
So some of the challenges we face, so I am not a plant bioinformatician. I work mostly with humans and animals and pathogens, so it was very new for me to work on plants, but Africa BP has been really great in, linking me with all the right people, because you don't know what you don't know until you realize that there's more to know. And another challenge that we specifically faced was computing resources. So because the plant that I was working with was so complex, a lot of the typical resources that we throw at assemblies wasn't enough to resolve this complex genome. So I'd had to go, just speak to other African researchers and to see how they do things and how I could improve how I was doing it. And yeah, we've made some good connections with thank God's team as well. I think the Sadiq has been very helpful, and I've learned a lot through the whole process. I'm just very grateful and happy that we can sequence African plants on African soil and analyze it ourselves. Over to you, Michael.
Yeah, I think I would say the same. The complexity of the genome. For my case, I've worked on cassava. For some reason, it was a bit straightforward having the reference of cassava. But this one also it's a new genome that has not been sequenced before, so there is not much reference to compare. And even like you go high on the family level, is then we get species that have been sequenced. So there was not much to like compare. So you actually starting from scratch, and the complexity of the level of duplication that was really sort of challenging. But of course, with the network, Sadiq also really helped in the discussion. Thank God to within the African Assembly group, So this was really helpful. And also just to mention, like I'm really completing the last year of my PhD, so a lot going on as well. So then this was really I think a lot of work, and hopefully now we're getting to the end of it. So this is really nice. And actually sequencing, I really like the idea, thank God, like sequencing in Africa, doing it in Africa, collaborating within Africa, and building capacity within Africa. So that's really nice. Yeah.
Yeah, thank you, Michael and Ansia. I think this has also been very interesting because it allows us to see firsthand what the challenges are, and also allows us to see and experience firsthand how we can actually drive and move things. I mean, looking at what you guys have done in this genome, I was telling someone the other day, they said, "Oh, thank God you called me in." I said, "No, I think the genome is now on autopilot in the sense that we now need to publish it." So everything, you know, so it's actually been very fantastic. And what awaits you guys now is how do you now take those bits, and then after it's published, take those bits and also um, uh, uh, increase it in a geometric fashion in terms of knowledge, in terms of giving to people, in terms of, making it open, giving people the opportunities that, the experiences and sharing it, especially on the African continent, because one could sequence all this. There are institutions outside Africa that can sequence all this in a year or 2 years, but we said no, we don't want that. We want to sequence it, and I've seen people who have also built their opportunities by just doing it because you have that sense of ownership, confidence, and things like that. So I'm really pleased with this. So there are also many of those opportunities and projects within the Africa BP also in that same direction. And most of the time, I think to myself, what if we can scale this beyond just the number we have? What if we can take it times 10, times 20? Then that would then get us closer to the Africa we want, which all of us on this call, I believe, do want. And also that will also increase the number of researchers, for instance, because Africa has the lowest number of researchers globally. An example is that the average number of researchers per million people is around 1,400 researchers per million globally. But Africa has just around 300, 350 per million people. So which means the number of people that can actually drive things low. So we need to then increase it. And I think that is one of the significance of a project like the African BioGenome Project. And I really want to thank you, Ansia and Michael, for also showing that this can be done. And I hope that this will also provide a sense of motivation and inspiration for everyone on this call. This is a high quality assembly, high quality activities, and lots of things that experiences shared over the past few months. All right,
I think I want to just say that I think we shouldn't give up because from what we have seen is that the project started a bit slow. Ansi, I think you also experienced that slow start as with Michael, a lot of retries, and thank you for not giving up because eventually it worked. And that's now we're looking forward to, implementing that scale globally. Yeah, thank you.
Yeah. And I think you guys didn't give up because at some point you will say, "Oh, like, okay, all right, that's it, right?" So, what especially with all of this deploy polyploid, you already is just seeing the next species and doing something with it. That is where the whole thing is. All right, thank you, and Professor Ann. This was supposed to be a comment, but then it's turned into a panel session. And all right, thank you so much, everyone. Michael and Ansi, I hope you still hang around. We have a few more, one or two more presenters, and then we can wrap up for today. Do we have we have people on site? Do we still have control on site? Professor Julian, do we still have control on site?